This is episode 9 of the series, Organic Chemistry for Biohackers, and today's episode is all about redox reactions and iron absorption. So if you want to know how your body actually absorbs iron, or how free radicals and reactive oxygen species are able to oxidize cholesterol or oxidize DNA and drive DNA mutation, you're gonna want to know what the frick is even going on with redox reactions. So without further ado, Episode 9 of the series Organic Chemistry for Biohackers, Redox Reactions. Part 1, let's define redox reactions. Redox reactions are all about electron transfer. So in every single redox reaction that's going on, one atom or molecule is being reduced and one atom or molecule is being oxidized. These are not independent events. They cause each other to happen. So for something to be reduced, it must gain an electron. For something to be oxidized, it must give up an electron. When a molecule is reduced, it gains an electron, and recall that electrons have a negative charge. That's why we say it's reduced. This atom's charge was reduced, it decreased, it got more negative when it gained an electron. When a molecule is oxidized, it's losing an electron. So in our redox reactions, some molecule is oxidized and it is losing its electron, and this electron is going to the other atom or molecule and becoming reduced. The reason why we say the molecule losing the electron is becoming oxidized is because in biology or biochemistry, oxygen is a very strong oxidizing agent. What oxidizing agents do is they cause oxidation. Same thing for reduction, reducing agents. Reducing agents cause reduction to happen. So if oxygen is acting as an oxidizing agent, he is causing some molecule or atom to lose an electron. If iron is acting as a reducing agent, iron is causing some atom or molecule to become reduced, and iron is therefore oxidized in the process. So an example could be Fe2+. Fe2+ could be acting as a reducing agent, and when he acts as a reducing agent, he's giving up an electron and he's becoming Fe3+. So if Fe2+ is acting as a reducing agent, he's giving his electron away to someone else, and he himself is becoming oxidized. So let's just make this super clear and concrete. Reduction is the gain of electrons. Reducing agents cause something else to become reduced, and they themselves are oxidized in the process. Oxidation is the process of losing electrons. Oxidizing agents, like oxygen, will steal someone else's electrons and thereby in the process become reduced. Part 2, we are going to go over biologically relevant oxidizing agents and reducing agents. So your biologically relevant oxidizing agents are oxygen, ozone, and hydrogen peroxide. Because they are oxidizing agents, they are causing oxidation. They are causing some other molecule to lose electrons. Or you could think of these guys as just being like really nasty and rude and just stealing electrons. However you want to think of it, what you need to know is that oxidizing agents, oxygen, ozone, hydrogen peroxide, these are stealing electrons and causing oxidation. Let's focus on oxygen acting as an oxidizing agent because this ties back into the video on free radicals and DNA damage. So when oxygen is causing oxidation, oxidation is the loss of electrons, oxygen is stealing electrons, and when he steals those electrons, he himself becomes reduced. So oxygen plus an electron becomes superoxide anion. Superoxide anion is a reactive oxygen species. It is very unstable, it is highly reactive, and it causes a huge amount of cellular damage. It specifically can be oxidizing DNA. So superoxide anion can oxidize the DNA-based guanine and damage it, turn it into 8-oxoguanine. 8-oxoguanine pairs with adenine instead of cytosine, and if that cell replicates DNA with the oxidized guanine pairing with adenine, one of those daughter cells is going to be mutated with an AT pair instead of a CG pair. If none of that makes sense, but you're really curious about it, watch my video on free radicals and DNA damage, because literally that whole entire video was all about the DNA-based guanine being oxidized. Superoxide anion, huge disaster case for your cell. It is extremely unstable because now it has this extra electron. Having that extra electron right there is what makes this so biologically bad, because now superoxide anion is going to travel around your cell and he's going to want to get rid of this electron. He's going to show up to lipids in your cell membrane, show up to DNA, proteins, or like the myelin sheath in your neurons, and he's going to dump this electron onto those good healthy molecules. When he's dumping this electron onto good healthy molecules, those molecules become reduced. He's happy again because now he's gotten rid of this electron. He's just normal oxygen, but now some other guy over here that was having a good time got that electron and became reduced. Now he's unstable. So he's not having a good time. This can also initiate something called the radical chain reaction, and the radical chain reaction is exactly how it sounds. It is a chain reaction of redox reactions. So superoxide anion had an electron, it dumped that electron onto some other molecule. Superoxide anion lost its electron, so it became oxidized, but the guy over here that he dumped his electron onto just gained an electron. He became reduced. Now he's angry, upset, and unstable. He doesn't want this electron because now he's highly reactive and unstable. So he's going to dump his electron onto some other molecule. He's going to be oxidized. He's gotten rid of that electron. He's happy again. But now some other guy over here just got dumped an electron onto him, and now he's unstable, highly reactive, and not happy. And this radical chain reaction will literally just propagate. It just goes on and on and on and on and on until some antioxidant shows up and neutralizes the free radical situation. So those are biologically relevant oxidizing agents. One more thing, actually. Hydrogen peroxide causes lipid peroxidation, so it is oxidizing lipids. It can be a cause of oxidized cholesterol, and it can be a really big disaster for cell membranes and cellular health. So all your cells have a lipid bilayer, phospholipid bilayer. This is like a nice, plush, literally fat membrane, lipid membrane, keeping your cell safe and happy. This lipid bilayer is critical to cellular health and cellular function. When hydrogen peroxide is oxidizing the lipids of your cell membrane, it's literally just going like zap, 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 burning holes in your cell membrane, and it can cause your cells to lyse or burst. If your cell bursts, it's literally just spewing its guts all over the place, and game over. It's cell death. So this can cause apoptosis and cell death. This can cause DNA damage, DNA mutation. This is just not a good thing you want to happen in your body. One caveat though, your immune system will use free radicals. Your immune system will do this thing called a respiratory burst, and that is when it is using a bunch of free radicals to kill bad bacteria. So in most cases, all these oxidizing agents are causing disaster in your body, unless it's your immune system and it is done in a very controlled manner to knock out bad bacteria and knock out invading pathogens. Part 3, biologically relevant reducing agents. And if you are interested in health and metabolism, if you take your B vitamins, you're going to be very happy about this example because we are dealing with NADH and FADH2. These are both critically important to metabolism. So NADH shows up in glycolysis, the process of burning sugar, and NADH and FADH2 both show up in the tricarboxylic acid cycle, which is another process of burning fatty acids and burning sugar to make energy. NADH and FADH2 are both made from B vitamins. So NADH is nicotinamide adenine dinucleotide. The N part is made from the B vitamin niacin. If you are deficient in niacin, if you are not getting in enough B vitamins, you don't have this N here. You can't make this molecule, and metabolism and cellular health and energy production really suffers. If you're not getting enough riboflavin, you cannot make FADH2. Deficiencies in riboflavin will also impair glucose metabolism, fatty acid metabolism. So NADH and FADH2, these are two very metabolically important reducing agents. What NADH does is it reduces things, but what it actually does specifically in metabolism, NADH shows up at the electron transport chain and it dumps electrons onto complex 1 of the electron transport chain. When it's doing this, it is reducing complex 1, it's giving an electron to complex 1, and then it's also getting rid of this proton here. Let me show you what that looks like. NAD plus, this is what is showing up in the tricarboxylic acid cycle. This is NADH in its oxidized form. It doesn't have an extra electron associated with it, and in this case, we are representing the reduction by the transfer of a hydrogen ion. One teensy detail I forgot to go over is that we can also show reduction and oxidations taking place with the transfer of hydrogen atoms. So a hydrogen atom, just an H, has 1 proton and 1 electron in its nucleus. When this proton combines with this electron, the positive charge on the proton is canceled out by the negative charge on the electron. These charges cancel each other out, and we get the hydrogen atom. So in some oxidation reduction reactions, instead of just straight up showing the movement of an electron, we are actually instead showing the movement of a hydrogen atom. That's what's going on in the case of NADH and FADH2. NADH, this is in its reduced form because it has this hydrogen atom here. FADH2, this is in its reduced form because it has an extra hydrogen atom. So NADH in its oxidized form is NAD plus. FADH2 in its oxidized form is FADH. NAD plus oxidized, NADH reduced. FADH oxidized, FADH2 reduced. Part 4, redox reactions in the context of metabolism. NAD plus in its oxidized form is going to show up to the tricarboxylic acid cycle and it is going to become reduced. So in the whole entire tricarboxylic acid cycle, we are starting with a molecule of citrate and we are just processing it through all these enzymatic reactions. And the goal of these enzymatic reactions is to extract high energy electrons from citrate, and citrate we ended up getting from glucose in glycolysis. So it goes glucose into glycolysis, then you get pyruvate, you process pyruvate into acetyl-CoA. Acetyl-CoA combines with oxaloacetate to make the molecule citrate, And that, once you have that molecule of citrate, you've officially started the tricarboxylic acid cycle. In the tricarboxylic acid cycle, NAD plus oxidized is hanging out in there, and it is waiting to scoop up electrons. It picks up those electrons in the form of a hydrogen atom. So we represent that as NAD plus a hydrogen atom, or we could represent that as NAD plus one proton plus one electron. And recall from literally like 24 seconds ago, one proton plus one electron equals hydrogen atom. These two things show the exact same thing. NAD plus a proton plus an electron, or NAD plus a hydrogen atom, both of those make NADH. This NADH is a high energy electron carrying molecule, and it is carrying these high energy electrons from the tricarboxylic acid cycle, from this metabolic pathway of sugar or fat oxidation, and it is taking that high energy electron and delivering it to the electron transport chain. NADH is a reducing agent because he himself right now he's reduced, so now he has the potential to reduce another thing. He specifically reduces complex 1 of the electron transport chain, and when he reduces complex 1 of the electron transport chain, he turns back into NAD+, right? So he's a reducing agent. He caused the reduction of something and thereby became oxidized in the process. And now he just floats on back to the tricarboxylic acid cycle, and hopefully we are still burning sugars or fats for energy. And this process just literally goes on and on and on and on and on until either you die or the cell dies. But this is what's happening with NAD, NADH in metabolism and redox reactions. This is super critical for energy production. Literally, without any of this happening, you're gonna have a huge problem of metabolic dysfunction. Your cells are not gonna be able to produce enough ATP, and this is really not a good thing to happen. You need energy for literally like everything going on in your body. This is super important. Also, by the way, NAD, nicotinamide adenine dinucleotide, niacin, the B vitamin, you need that B vitamin present to make NAD+. So eat good, healthy food. If you need to supplement, figure out what supplements you need to take, but you can get a lot of high quality B vitamins, high quality nutrients from literally just having a good diet. Salmon, bison, venison, eggs, these are all really good sources of B vitamins, and these are all going to be helping you out with your metabolism, with your metabolic health. This concludes part 1 of the video on redox reactions. Part 2 is going to be all about iron absorption and iron bioavailability. So now that you've learned all about redox reactions, you can understand what the frick is even going on when we're talking about iron absorption and vitamin C and iron bioavailability. If you got any value out of this video, like, comment, subscribe. That helps me put more resources, time, and energy into making these videos for you. So I hope you all had such a lovely day, and I'll see you in part 2, which I am literally about to film right this very second. So see you later, everybody. Bye-bye.